Dear student, course number GPB 311 क्रॉप इम्प्रूवमेंट खरीफ। इस कोर्स के अंतर्गत हम पीजीएनपी ब्रीडिंग पढ़ रहे हैं। आज के वीडियो में हम पढ़ेंगे ब्रीडिंग ऑब्जेक्टिव्स फॉर पीजीएनपी। So let us start. Breeding objectives. Like other food crops, the breeding objectives for PIGEONPEA are in common with some specificities as per requirement of the crop. Number 1. Breeding for higher yield. Major traits for priorities for breeding higher yield in PIGEONPEA are days to 50% flowering, days to 75% maturity, plant height, branching pattern, number of pods per plant, number of seeds per pod, pod length, 100 seed weight, that is test weight, and ultimately yield per plant. These all are yield contributing characters which should be taken into consideration for breeding varieties for higher yield. Number 2. Breeding for specific maturity groups. As we know, PIGEONPEA is a crop which has three types of maturity: early maturity, for medium maturity, and late maturity groups. So, breeding for early maturity varieties, that is the duration of 110 and to 140 days of maturity. B. Breeding varieties for medium maturity duration, that is in between 140 days to 160 days. 180 days. And breeding for varieties for late maturity groups. These are the varieties of the duration of 250 to 280 days. Number 3. Breeding varieties for disease resistance. As we know, it is very important. PIGEONPEA suffers from three important diseases in Indian subcontinent. These are wilt, which causes loss up to 25 to 20% yield losses; sterility mosaic, which losses yield up to 5 to 10%; and phytophthora, which causes yield losses up to 10 to 15%. First, we will take wilt. It is caused by Fusarium udum. Wilt-resistant varieties or genotypes or lines should be included in breeding program like ICP 8863, ICPL 87119, that is Asha, BDN1, BDN2, that is Maruti, Narendra Arhar 1, ICPL 87, that is Pragati, KM7, that is Jawar, TTG, or ICPL 87051. These are the lines which should include in the breeding program for wilt resistant varieties. Number 2, sterility mosaic. The causal organism of this disease is still unknown. It is spread through mite vector. Escherichia cajunii. The varieties resistant to sterility mosaic are Bahar, GA11, that is Sarad, Narendra Arhar, ICPL 8705, ICPL 87119, that is Asa, and BSMR 736. These all genotypes or varieties should be included in the breeding program for developing a variety of sterility mosaic. C. Phytophthora blight. This disease is caused by Phytophthora drexleri species Kejeni. This pathogen is highly variable. None of the released varieties is still resistant to this Phytophthora blight. However, some breeding lines are reported to be tolerant, not resistant. Are GAUT 82-55, KPBR 80-1-4, GAUT 82-104, ICP 8103, KPBR 80-2. So these all should be included in the breeding program for the resistant to Phytophthora blight disease. At the ICRISAT, standard field screening techniques against these three important diseases have been developed. The sick plot technique is developed for fusarium wilt. The technique consists of uniformly incorporating all dead plants debris into the soil. Such a plot is then ready for screening. During the screening, every third row is planted with a susceptible cultivar. These rows serve as check and helps in the monitoring as well as maintaining the wilt sickness of the plot. To screening breeding material simultaneously for resistance to these three diseases, multiple diseases screening techniques is in used at ICRISAT. Number 4. Breeding varieties for resistance to insect pest. In India, pod borer, Helicoverpa armigera, and pod fly, Melanogromyza obtusa. Both are most important pests of pigeon pea. For screening against these insects and using identified donors is an important resistant breeding program. Usually field screening techniques are used. In this, test material along with controls are exposed to adequate number of insects under the pesticide free conditions, and development or multiplication of insect and the damage caused on the plant are compared. A few pigeon pea lines reported to be resistance against these two insects are for pod borer ICPL 87088, ICPL 87089, number 148, HY2, Fule T1, Fule T3, BDN1, BDN2, PL 8996. And for pod fly resistance, the genotypes are 11012-2, MA2-3, SL12-2, SL42-3, ICP 3615, ICP 4745. These sources could be used in breeding program using pedigree or bulk approaches. Reducing the duration of crop may minimize the damage caused by the pod fly. It is the management concept for resistance to insect pest. Number 6. Breeding pigeon pea varieties for abiotic stresses. Major abiotic stresses in pigeon pea are drought, waterlogging, extremes of temperature, frost injury, and salinity. Effective screening techniques have yet to be perfected for these traits. ICPL 227 is found to be tolerant to salinity. Next, breeding for quality traits. Grain quality refers to nutritive value, the presence of anti-nutritional factors, dehulling quality, cooking quality, consumer acceptance, seed size, shape, color, structure determines the important traits for market or consumer acceptability. Two sulfur-containing amino acids, methionine and cysteine, are limiting in almost all pulses, including PIGEONPEA. Anti-nutritional factors, trypsin inhibitor and amylase inhibitor should take to consider by the breeder. And next point is breeding for specially traits. Fresh green pea from green pods are used as vegetable in some parts of Gujarat and Maharashtra. There is need to develop such type in early and medium maturity groups, breeding for higher photosynthetic efficiency with reduced respiration losses and for reduced foliage to improve the harvest index are the characters to be taken into the consideration. So these all are about the breeding objectives for pigeon PE. Thank you.